Okay, so welcome to the April webinar of PAASTA. So this is the Paleoproteomics and Archaeology Society for Techniques and Advances. So the purpose of these webinars is to inspire connection and communication between members of the community. So please feel free to reach out and ask the speaker questions at the end of the talk. However, the webinars do contain published or also unpublished data, and so the resulting conversations and the content should not be shared without the express permission of the speaker. And please note that these webinars are not formal presentations, and so the speaker should be contacted directly if you're seeking interviews or statements for reporting. And finally, we want to encourage a very safe and positive space for the sharing of knowledge and information in the community. So we ask everyone to abide by the code of conduct, which I will post a link to in the chat. And so this week we are very happy to welcome Johanna Krueger, and she is going to be talking about how robust is a phylogenetic inference from limited protein sequence data. And I've also popped a link to her preprint in the chat as well. So Johanna, if you want to start.
Sure. Thank you, Helen. I hope you can all hear me well. It was a little bit cut when I heard you, Helen. I don't know if it's your connection. Is it fine?
Yeah, I can hear you very well. Was there a problem hearing me? I can hear you very well. Yeah.
Okay, perfect. Yeah, hello everyone, and thank you for coming to my talk about the robustness of phylogenetic inference from limited protein sequence data. By limited protein sequence data, of course, I mean paleoproteomic data, and we're gonna explore this at the example of primates. I'm Johanna Kreuger from the Comparative Genomics Lab of Thomas Marcus Bonet in Barcelona, and yeah, let's dive into the motivation, what motivated us. So ancient biomolecules have been used for molecular evolutionary studies for quite a bit. And I just wanted to take a quick look what we can do actually with ancient DNA. Some of the oldest, to my knowledge, is mammoth DNA, and there have been quite a bit of studies. For example, there's one from Diaz de Molino, where they looked at 16 mammoths between 12,000 and 700,000 years. And just the amount of information you can get, I mean, this is a very nice sample, was over 1 million high-quality fixed variants that were just unique to the woolly mammoth that they studied. And over 3,000 genes contained non-synonymous mutations, so there was change in at least one amino acid. Now, if we want to study mammals and in deep time, one method that I'll focus here is usually the use of tandem mass spectrometry, and one popular tissue is tooth enamel. It is because tooth enamel forms crystals that trap the proteins very well, and it's very hard tissue, so it survives very long in time. It has a very small proteome, and the proteome is already fragmented in vivo. It's part of an enzymatic cleavage process. And the age ranges of what is published right now is in the ballpark of 2 million years. There are older samples that work. You will also see that later in this presentation. But the majority of what is out there is between 1 and 2 million years. So I took some studies on tooth enamel sequences just to see how much of all 10 proteins combined you could possibly get. And the amount of amino acids that were covered range from 450 to 1,000. So on average, it was 745. And on average, 1.3 derived variants that were different from the reference species. They're often called SAPs, single amino acid polymorphism. It would be probably better to call them SAS because they're a substitution when we talk about difference between species, but that shouldn't bother us for this presentation. And nevertheless, this data has been used for phylogenetic application. An example where this works fairly well is here from Welker et al. Gigantopithecus, which we see here is a pongine, so it belongs to the family of orangutans, and even divergence times have been estimated. And the data, just as a reminder, again, it consisted of 6 proteins that they could find. They made multiple sequence alignments, concatenated them. There's 456 amino acids in total, and 3 variants that were derived and exclusive to Gigantopithecus. And this kind of studies, ironically, it brings us back into the old days. And what I mean by that is that protein sequencing actually preceded DNA sequencing. And so it happened that some of the first studies on molecular evolution were made on protein. It was horse cytochrome C in the 60s here. And what we see here is also just pairwise comparisons, seeing how they vary, which amino acids differ between these pairwise comparisons, and bringing that into relation with divergence time. But how reliable are these phylogenetic estimates? So luckily today we have high quality whole genome DNA data to corroborate our findings, which people back then didn't have. And so we wondered, what if we just simulated ancient data from modern genetic data? Well, and this is exactly what we did. And by we, I mean mainly Ricardo, my co-first author, who's also here, who did a whole lot of the hands-on work, a really big part. And Esther, who supervised us, who is in our lab. And the data that we use is mostly, not exclusively, but mostly from this publication from our colleague Lucas Codena of 233 primate species of over 800 individuals. And this is more or less half of the primate species that are out there. So we used this genomic data to predict proteins, and we created different sets of enamel proteins. I'm going to briefly introduce them, and the differences between them are going to be important later on. So we came up with the first set of proteins, of 5 proteins, that when I looked back into the studies that I presented earlier, kind of always recovered. So they are a rather safe bet that you will find them. There's a larger set, which I call the theoretical enamelome, that if you see that in some publications or in some literature, you will find those. Now, later we discovered that tuftilin, actually, despite being mentioned in the literature, I couldn't find any experimental evidence in the literature, nor in my own data. But at that time, we didn't know that. And then there's a larger set, and this includes collagens. Now, collagen 17A1, which we see here at the far right, is known to be actually part of the tooth enamel. The other 3 collagens are thought to be part of the neighboring tissue in the tooth, which would be dentine, and that if they pop up in your paleoproteomic experiments on tooth enamel, they're most likely some kind of contamination that comes from the dentine. The contamination can happen, especially if you have very old samples. So you have to imagine you kind of have, it's more a rock than anything, and it's really hard to separate that, especially if you have very thin enameled species. So it can definitely happen, and it's also very brittle. So risking to clean it is also risking to break your enamel chip. So one of the first questions when we had oral proteins bioinformatically predicted from the DNA, we aligned them. We wanted to know where's the variation in them. And this is exactly what we were looking at first with Shannon entropy, which is a very simple measure. And we could already see some differences, that the collagens are rather conserved. But we were also looking for a measure that is maybe more evolutionary aware, so that amino acids like leucine, isoleucine, that are kind of chemically similar to each other, but also mutate easily into each other, that this is accounted for. So we used right for site, and we kept using right for site more for downstream analysis to assess the variability. And we can see very clearly here that collagens, it's been said before, but here we could clearly see that collagens are especially conserved. So the whole enamel proteome is known to be rather conserved in comparison to other proteins, but especially collagens are very conserved. And we can also see other interesting features that are known about these proteins. They're usually not too well studied, but we know, for example, that enamelin here, it has this 32 kilodalton fragment that is a cleavage product. and that seems to play a key role, and it's very conserved. And so are parts of the enzyme that cleaves it, MMP20. So one of these bumps down here is the active center, and another one are some phosphorylation sites, for example. So from these concatenated alignments of our proteins, so basically you can imagine all the sequences put together into one large sequence, into one large alignment, we made phylogenetic trees. And we compared these phylogenetic trees back to a reference tree. Our reference tree is the tree that stems from the publication, where also most of our data stems from. It is made from ultra-conserved DNA elements and from high-quality genomes, and it is fossil calibrated. And we have rather high confidence that this is close to the actual phylogenetic tree of these primate species. And we assessed the difference between our trees and this tree with the Robinson and Folds distance. So for those of you that don't compare trees maybe too much, Robinson and Folds distance can serve to compare two trees. And basically, it counts the amount of steps that you would have to do to transfer one tree into the other, to transform it into the other. And we see here we have our 3 sets of proteins, the 14 with the collagens, the 10 and the 5 that usually we always get. And we also tried 2 methods, maximum likelihood and Bayesian. The Bayesian shouldn't bother us too much here. It turned out to be very similar to the maximum likelihood, so we were just more confident in our method. And if we look at this, we have a Robinson and Folds distance of 108 between our tree with most proteomic data and DNA data. So to understand what this means, I looked into the trees and manually expected them. I'm not going to show all of this here, but what I could basically see is that at family level, everything for all of the trees worked out pretty fine. It was very consistent, and even at genus level, the placements were in accordance with our reference tree, which was a good result, I think. And only the arrangement of the species within a genus, for example, here we have a lot of macaques and their order might differ. They may not be distinguishable with protein data, so that may change. So this causes this. So that I can say for any further slides that you see that 100 is more or less a baseline where the trees are as close as they can get to each other and where all the genera are more or less in accordance with our reference. But one thing happens if we use the most complete tree that gives the best result. It includes collagens, as I said, this data set of 14 proteins, and then something very interesting happens. So we see here a tarsier, and these little things, they look like lemurs or lories, but they are not. They actually, if we have our DNA tree here, it's one of the deepest splits. And traditionally, they have been placed because of their looks of their morphology with lemurs and lories. But now we know with modern morphological data and especially with whole genome data that they most likely belong to the anthropoids. This is consensus right now. And then when we made our protein tree, though they happened to cluster again with the strepsirrhines, so with the lemurs and lorsies. I mean, we cannot see this here, but this actually they share one root together. So that was a curiosity for sure to see with our data, and it was surprising because it was the most complete data set we had. We don't know exactly what causes this, but Ricardo did a lot of research in which proteins are driving this. And it's not enough to have just one collagen or 2 random collagens. It always needs a particular one, I think collagen 1A2. And collagen 1A2 itself also not. It needs a combination of collagen 1A2 with at least another one. So there's something, some conditions needed to tip the scale, so to say, to change this placement. So that was one of our first important takeaways, that if you have more data, sometimes some proteins seem to have their own evolutionary history or other reasons that they can change these deep splits. But of course, one of our core questions was, what if we made phylogenies based on degraded samples? So we somehow needed to simulate data that comes from fossils. And basically what I did is I deleted columns. So this represents my alignments of my 14 proteins. And I just deleted columns. And the older the sample I wanted to simulate was, the more columns I deleted. And I tried to do this in an informed manner. So this is still far from having statistical support because there's simply not enough experimental data for every protein, for every age range. But still, I try to grasp whatever is out there and make use of that and look at that and let it inform our downsampling. So here's just one brief example of the protein amyl X. I used the modern human as reference and aligned everything I could get from Homo antecessor, Paranthropus, Gigantopithecus, horses, and so on. And also to very old samples. So you see already, as I said, most of the published data is between 1 and 2 million years old. And we added from our own lab an horse-like Hippotherium and an elephant-like Dinotherium. Those are both 10 million years old. And we still could recover a little bit of data for those. So that gave us an idea which parts of the protein is preserved for the longest. This is one of the outcomes. So first, our model for samples that are somewhat degraded, but let's say the least degraded. And you can see that we have, we recover, for example, the part that is very conserved in enamelin. So that is interesting to see how conserved or variable actually the parts are that across the experiments that have been published so far, how it coincides together. You can see here, and you may wonder why I have so much data of the collagens. So we try to be as pragmatic as possible so that it can inform future experiments. And usually at that age, I found a lot of data that was derived from bones because collagens are in bones. And it was rather good data. It was rather complete data. So I assumed that if you had a sample that young, you could make use of bone data. Bone proteins don't preserve as long as enamel, but again, if you have such a young sample, you could make use of this. This is why we included it, and we hope, of course, that this would be an interesting case for people that work more with collagens. For the older age, then you can clearly see that it goes more to the typical 5 proteins that are always recovered. And if you go even further, I made it less. So for 5 million years, I had to anticipate a lot between the 2 million year old samples and the 10 million year old because there's not a lot of data out there. And finally, for the 10 million year old sample, a couple peptides for collagen and enamelin, but mainly amyl X, which is by far the most abundant protein in an enamel matrix. So we took these alignments, of which we now just chose certain columns, and again created phylogenies from this, and again compared them back to a reference. So for this, for now, only the leftmost column is interesting for us. We got Robinson Folds distances, and they were not as good as the full sequence, but still at this fragmentation stage of 100,000 years, the family level was still okay because we had so many collagens. Again, the tarsiers were placed in the wrong place with the stress strains, but anything else was rather robust and gave us rather good results at family level and even mostly at genus level. This is also more or less true for 1 to 2 million years. I just checked the tarsiers place wrongly, but in a different way. They're now the rotary thing, but anything else is more or less fine, except for, you can see how it starts to crumble here, that chimpanzees, which are the sister group to modern day humans, now are considered the sister group of gorillas, which is incorrect, as we know. So we could expect mistakes at this magnitude, if you want to say so, if you have data that is as fragmented as this. At 5 million years, most of the signal is lost. And at 10 million years, the tree basically loses all of its structure. But of course, in real life, in a real experiment, you wouldn't have fragments of many species at one time. What you'd rather have is you have your species of interest with a couple individuals that have fragmented data and a reference that is modern. So you would have the whole sequence. And this is exactly what we tried to study. We made a couple of case studies. I'm just showing one of them here. So we just pretended, what if chimpanzees were a fossil? Let's say in our 5 million year old model, and we fragmented it. So we had the full sequence data, and Ricardo sampled it down a lot. And we made a phylogeny. And as I just mentioned, at this stage, or even at the 2 million year old stage, chimpanzees here, Pan-troglodytes, clustered with the gorillas wrongly. But if it's only the chimpanzee that is simulated to be ancient, it goes again correctly with the bonobos together here, together with a human. And we also wanted to know, since we had good data for reference, how good are the divergence time estimates? So I did this with the help of Robin Beck, who also helped a lot, or was guiding the divergence time estimates of our reference trees via fossil calibration. And what we can see here first is that the intervals that I estimated from our protein data overlap with the intervals here from the DNA data. In general, the estimates based on the protein data were in their mean 21% younger than reference, which isn't too surprising because the protein data, as I said, it's highly conserved. It doesn't show too much variance that could inform deeper divergence times. Just one interval did not overlap. So between the orangutans and all the other great apes, I got a younger age, whereas the DNA data gave an older age and the intervals do not overlap. We think that there must be some signal in the DNA data that makes us way older, because what I got here is pretty close to the fossil calibration. So it does not really seem to be a product of my proteins, but rather there's something in the DNA that informs this estimate that is simply not captured or reflected in our super small proteome. Another interesting thing was that our chimpanzee here, Pan-troglodytes, the split was estimated to be a bit older than that of the reference. I don't know exactly why this happened. I think there's some kind of randomness in what you catch experimentally that can, if you have so little data, drive this, but nevertheless, the confidence intervals overlapped. Next, we were interested in which sites drive the phylogenetic placement. Now, this may seem a bit obvious that in the super conserved proteins, it is the variable sites, but it was still good to explore this since the reference data, the DNA, is based on ultra-conserved elements. So we tried to divide our data into variable columns and columns that seem rather conserved. We used channel entropy and rate for site as the measure. We normalized it to a value of 1 and then just defined everything that had a value of 1 as variable and everything that was below as conserved, and did the same here. So Ricardo picked these columns that fell into these areas, combined them, made new trees, and compared them back to the reference tree. And what we see here, again, we will only focus on the uppermost row, is that whenever we took the variable sites of our alignments, you see these tiles are lighter, so the Robinson and false distances are less, so they're more similar to our reference tree. And very interestingly, for rate for sites, the distance is 108 between our tree from proteins and the reference tree. And this is exactly the same value, the same distance that the full sequence tree has. So in this case, it doesn't really matter if you take the full alignment or just what rate foxide defines as variable. You get pretty much the same quality of output, if you want to say so. And one last question that I was thinking at, if we apply this data set, this proteome, to phylogenetic inference, is if the loci are independent enough. So in genetic studies, you have to make sure that if you want to do phylogenetic analysis, that what you look at and what you combine has some kind of independence. Otherwise, you could be biasing towards the evolutionary history of one specific locus or one specific gene or cluster of genes. And this is exactly what we have here. We have a cluster of genes. So these proteins in humans, but I also checked, it seems to be the case for all the mammals I've checked so far, just in other chromosomes, they cluster together. Enamelin and ameloblastin even are the offspring of a gene that has duplicated of the very same gene. And so is AMEL-X. So AMEL-X, enamelin, and ameloblastin share a lot of similarity to each other. And AMEL-X, again, has the paralog AMEL-Y, which many of you know is very useful for sex determination, since they are both expressed if you are male. And they share some similarity to each other. They do not combine. So usually in meiosis, when the X and Y chromosome meet and they have a region where they could recombine, AMEL-Y is not in that region. And yet I have seen in my own data and also read that there seems to be something going on. There's some relationship that keeps these similar, like these sequences rather similar to each other. People have discussed if it could be gene conversion or other things. And then there's, of course, the collagens, which are in different chromosomes, but they are still so similar to each other. So just in humans, they can share up to sequence 70%, 72% sequence homology or identity, I think. So to measure the dependence of these genes that I just presented, I got the help from Jordan Little, who's supervised by Nathan Clark. So Jordan Little has been improving the ERC scores. So ERC stands for evolutionary rate covariation. And basically what they do is they have a defined set of mammals. They have all the genes you can imagine. They're like phylogenetic trees for all of them, and see how the evolutionary rates between those genes covariate. So then you can get relationships between two genes. It is explained on their website. If you want to know more about that, I highly recommend it. It's also very easy to use. And we could clearly see here that AMEL-Y and AMEL-X, that their relationship stands out. So it was good to see it somehow quantified that between these two, there's a clear relationship. So we didn't have AMEL-Y in our data, actually, because the Y chromosome is often, it is very hard to assemble, and it's hard to get data from all of the primates, but also all of the mammals. But in this newer version of the ERC scores, they got the Y chromosomal data, so I could finally explore this. And it seems to be a very strong relationship, which may have implications because for now they are treated as independently. And another thing that we can clearly see here is the relationship of MMP20. That again is the enzyme that cleaves, for example, ameloblastin, or also enamelin, or AMEL-X. And there's clearly elevated values between those. And we can also see relationships here, for example, between the collagens, which share these high similarities, and also functional, functional interactions. What I don't know and what I'd like to understand more is the implications it has. Because despite all these dependencies that I just presented, it still seems to be the best to base phylogenetic analysis on all of these genes combined, so basically concatenating all of them to a large supersequence, instead of making trees on the single genes. I know there's people who look into this, but I think it needs a bit more attention. So to summarize what we found, meaningful phylogenetic inference is possible, mostly at family level and to a fragmentation stage of 1 to 2 million year old. Remember, the ages I didn't normalize them, but they are from temperate up to tropical regions. If you have better conditions, and with better conditions, I mean cold conditions, samples, this may change a lot. So it's rather the stage of fragmentation than the actual age. And also remember that if you have only one sample, which is experimentally more likely the case, that is ancient, and you place it with data from modern references, you can also get better results. The inclusion or exclusion of single proteins can change deep splits. We think that this may be not very often and a rather curious case, as we had with our tarsiers, but we surely don't know what is going on in other mammals, in other groups, if this can happen as well. And if it's a collagen thing, or if in other mammals, it could be other proteins. The estimation of the divergence times was surprisingly good for me, at least. It was better than my expectations. It was slightly younger, which I also expected. And I'd also like to try out more case studies, because I can imagine that this is also case by case. And some protein pairs display correlated rates of evolution. And if you know more about this, or if you have an interest in that, I'm very happy to discuss that with you. So what does all of this mean for you? Well, if you work with mammals, you can see that the patterns that we discovered for primates here in the lower look very similar as that to mammals, which we see here in the upper panel. We can see that the conserved fragment of enamelin is also conserved there. We can see that the collagens are very conserved, the enzymes. So there's reason to believe that what we found can be meaningful for other mammals. But in the end, I guess whatever group you're looking at, you would have to collect your own proteins, make your own phylogenies, see where the variation lies, check if you have any tarsiers, I mean, figuratively speaking, in it, some big derivations. And last but not least, sometimes still, especially with ancient data, we have to consult people that know a lot about morphology. They can sometimes be the only ones that can corroborate or contradict our findings. And with that, I'd like to thank you very much for your attention. I'd like to thank all of my co-authors, my group. Here we have a typical Catalan onion barbecue. I'd like to thank my funders. And if you have any questions later, you can send me an email, or now, just let me know. I'm happy to talk.
Thank you, Johanna. That was super, super interesting. If anybody has any questions, they can either just unmute themselves or put the hand up or pop questions directly in the chat, and then I can read them out. Oh, like you're getting applause. I do have one question which I wanted to start off with. So with the tarsiers, I mean, I know you just said at the end about consulting kind of morphological experts in like comparison with your data. For the tarsiers, is there any kind of like morphological weirdness happening with them and their like tooth development, or something about their enamel that is kind of putting them as a kind of outlier?
Yeah, well, I was trying, it was a rabbit hole and I had to stop myself. So morphologically, I mean, clearly they don't really look like the other anthropoids. And also in their enamel, there have been studies, but I don't know it from the top of my head, but there was something going on with their enamel as well, that the structure, it was an older study though, so I don't know what modern study says, but there was definitely going on something with their enamel as well. If that justifies what we found is hard to say, because sometimes I don't know if these proteins that we find themselves in the enamel are not the only ones that influence how the enamel is. I mean, when it's formed, there are whole cells that have more proteins. But it was definitely interesting. I don't know if anyone here is a tarsier expert, but it was one of my favorite aspects of the project.
Go ahead, unmute
Yeah,
yourself, Brad.
Yeah. Hi, Johanna. This is a great talk.
thank
I
you.
just had a question about how you, kind of simulate the degraded data, right?
Mhm.
So you're right that it's really hard to measure the robustness of, how you kind of put the whole thing to simulate the thing, right? So
Mhm.
do you, so was it just completely random selection of the columns to remove, or did you put any other criteria? So in terms of the the properties of amino acids, or the conditions where these proteins are found,
Mhm. Mhm.
or do you think
Um,
these criteria would matter, or is it just it's just complete stochastic random processes?
Yeah, it was rather the opposite. It was not completely random. It was maybe too stiff. So what I did is I looked at the data, and when I saw, okay, this is never recovered, this region is never recovered, even in the younger samples, I excluded it. When I saw that this region, I don't know if I have the yeah, I have it somewhere here, I guess. If I saw that a region was had here.
Yeah.
Yeah, for example, here, what we would see at the very right, this region has quite a bit of coverage. So I would include it always, whereas maybe this ones here at the beginning are a bit gap here. So I would have them in the younger models, but not anymore in the older ones. And it was the same set of amino acids um, for every individual. So it didn't go random selection. It would be nice to see something that is somewhat random,
Right.
but I didn't get to do this.
Yeah, I mean, obviously, one of the things that we've always been looking at is, if some domains of a protein sequence are susceptible to preservation, or if they are more susceptible to decay, right? And it
Yeah,
really depends on the artifact. It really depends on the site, and it really depends on the protein.
yeah,
So I was just curious, if you're putting any measure of probability as to if this particular amino acid is going to be, yeah, if this amino acid is going to be broken down or not. So this is a really open question. So I was just curious. So I mean, I've worked on
yeah,
experimental data, but as you say, we don't have a lot of this experimental data on collagen yet. So yeah, I suppose that would be a really interesting problem for from a phylogenetic point of view.
yeah, it definitely is. I hope the model can receive some more refinement
Yeah,
in the
definitely
years when more data is
more
out there.
parameters, yeah, more
Yeah,
attributes. I would say that you have to include more physiochemical properties of the proteins, and also the site specificity. I think this would also matter, because it just can't be completely random. We can't just attribute it as a complete stochastic process. So it'd be really cool to
yeah,
see how we can, yeah. But it's really cool what you've done. Yeah,
thank
thanks for
you.
the presentation. Cheers.
Pontus?
Yes, super interesting talk. Really great to hear the results. I guess I'm asking, so you compare to the sort of baseline species tree as the truth. And so I guess, is there kind of two things going on? One is the kind of partial case where the genes are unrepresentative, or the proteins are unrepresentative, and the other is kind of statistical uncertainty. So I guess I wonder if there could be cases where the tree does actually match more or less the species tree, but the bootstrap supports would be way too low? Like, did you do systematic looks at the bootstrap supports for your simulations?
Yeah, so everything I presented, except for, that's why I always said at genus level or at family level, we checked the bootstrap support. And that was actually also interesting because the error, for example, when we had the misplacement of the tarsiers, it happened with such a high confidence that if I didn't know any better, I would have accepted this split and would have trusted in it. Yeah, I don't know if that answers your question.
Yeah, I guess it's cool. Yeah, I guess it's going to be below the genus level, perhaps, where the bootstrap supports maybe are a bit more dodgy. Yeah.
Yeah. I mean, for the DNA data, it was fine. It was actually quite good. And for the proteins, the opposite happened because sometimes you have zero difference between them at a protein level.
I see.
Mm.
Thank you.
Do you have any further questions? Either you can put them in the chat or put your hand up. Okay. Well, Johanna, thank you so much for giving a talk.
Thank
It was
you.
really, really interesting, and it's given everyone lots of food for thought, I'm sure. So thank you very much. And we'll be sending around details of the next talk, which will be happening in May. And so, yeah, so hopefully see you all there for that. Bye. Have a nice afternoon.
Goodbye.
Bye.